Welcome to Chapter 8. Building on everything you've learned so far, we'll explore how DIA mode in HD Examiner Pro takes hydrogen-deuterium exchange analysis to the next level. We'll examine how DIA improves consistency by reducing manual bias and validating peptides automatically, the role of fragment-level populations in generating statistical confidence for Woods plots. By the end of this chapter, you'll understand how DIA delivers not only accuracy, but also deeper confidence in your HDX data.
Okay, so how does the MS1 analysis compare with the DIA mode in HD Examiner Pro? In the original publication of what was called AutoHX initially, the DIA method was compared with a completely manual validation mode of the same protein system. So this is phosphorylase B in a 6 time-point kinetics experiment. And the manual mode of analysis is compared with the automated mode of analysis, the DIA mode. And there are very, very, very little differences between the two methods. As you can see here on the right-hand side of the screen, we have individual kinetic spectra for each peptide, and the differences between the blue and the red line, which are the manual and automatic mode of analysis, are very, very minimal. In fact, in some of the automated analysis mode in the DIA space, the error bars might actually be a little smaller than in the manual case because we do have the extra MS2 dimension, and the software will choose to use either MS1 or MS2 based on which of the two creates the smallest amount of error at each time point. And that's how in automated mode you might have tighter standard deviations on your kinetics plots. Another comparison of the manual mode versus the automated HD Examiner DIA mode is looking at a Woods plot of a protein called polymerase theta with a drug named novobiosin. So this is the drug-bound state versus the non-drug-bound state compared in a Woods plot. The top view is a manual analysis of the system. So this is after going through every single peptide and validating the MS1 data, deleting any peptides that don't make any sense, and keeping the ones that are measurable. On the bottom side of the screen here, it's the same peptide, the same project, but now processed using DIA data. And so you can see here that the binding sites are actually quite similar on both analyses. So in fact, most of the Woods plot is very similar in both cases. You might see some differences, and some major differences are that there are a few peptides missing from the manual analysis mode. Upon closer inspection, it turns out that these peptides were misidentifications in the initial DDA experiment. So in DIA mode, they were not able to produce the expected amount of fragments to be able to verify their deuteration. And so in this mode, they were completely eliminated from analysis. The error bars on this plot represent the fragment populations coming from MS2 space. When we model the deuteration coming from all of the different subset of fragments, we can generate normal distributions. And the normal distributions, then we can plot their variances on the Woods plot directly to show you how much agreement there is between the final measurement and all of the different fragment populations in the data.
We have explored the fundamentals of hydrogen-deuterium exchange, examined MS1 and DIA workflows, and demonstrated how fragment-based validation can transform complex data into reliable insights. Throughout this process, you have seen how advanced techniques help overcome challenges such as overlapping peptides, enhance accuracy, and provide greater confidence in your results. By applying these approaches, researchers can accelerate discovery, streamline workflows, and achieve improved reproducibility in their protein studies. If you would like to learn more about how Connect and HD Examiner Pro can support your work, or if you have any questions about applying these techniques in your lab, we encourage you to visit our contact page. Our team of experts is ready to assist you in moving forward with confidence.